Welcome guys. In this session, we are going to discuss about the glycine amino acid. So let's first talk about the basic properties from the classification part. We know that glycine is a simplest amino acid. Simplest amino acid. If I just draw the structure of glycine, that is NH2C, COH, hydrogen and hydrogen. So you can see that the two valencies are having similar groups. So glycine does not have a chiral carbon. And once we are saying that glycine does not have a chiral carbon means it is optically inactive. That we have discussed in the classification part also, that it is the only amino acid which is optically inactive. It is the simplest amino acid, it is optically inactive. It does not have the chiral carbon. It is the non-essential amino acid. Non-essential amino acid. So these are the few points from the classification part. When it comes to there are two facts that you need to remember. Just facts, nothing much to go into that detail. If this is the structure of glycine, what I can say is in any amino acid we have two ends. In any amino acid we have two ends. One is the amino end, this is the amino end, and another is the carboxylic acid end. Carboxylic acid end. The amino end is also referred as the N terminal. And carboxylic acid is referred as the C terminal. So there are two ends that are there in any amino acid. N terminal, C terminal. Now what you do is, if you take the glycine and you add a methyl group, the molecule that you are going to get, because you are going to add the methyl group on the N terminal, this is referred as N-methylglycine. N-methylglycine. This N-methylglycine is also referred as sarcosine. So the point that I want to convey to you is, what is sarcosine? The point that I want to convey to you is, what is sarcosine? Sarcosine is just the other name of N-methylglycine. This is the point to be remembered. The next thing that you need to understand regarding glycine is, serine can convert into glycine. Serine is an amino acid from which you can make glycine. Now how to do that? To convert the serine into glycine, what you need to do is, you need to remove the beta carbon. Remove the beta carbon. If you remove the beta carbon from serine, the serine will convert into glycine. What is the meaning of beta carbon? The carbon which is next to the functional group is the alpha, followed by beta, followed by gamma and delta, so on. So if you remove the beta carbon from the serine, it will convert into glycine, and in that process we require vitamin B9 and B6. We require vitamin B9 and B6. To convert the serine into glycine, we require the B9 and B6. So these are the few basic points to be remembered for glycine. The function of glycine. There are various functions, and the first function is the creatine synthesis. Creatine synthesis. Creatine synthesis. Creatine is basically made up of 3 amino acids, and is asked multiple times that all of the following are component of creatine except. So there are 3 amino acids which are component of creatine, and they are glycine, arginine and methionine. So there are 3 components. There are 3 amino acids which are required to make the glycine, that is GAM. Glycine, arginine, methionine. Glycine, arginine, methionine. These are the 3 amino acids to be required to synthesize creatine. The second function of glycine, is to make glutathione. Glutathione synthesis. Glutathione is again made up of 3 amino acids: glycine, cysteine, glutamate. Glycine, cysteine, glutamate. Because it is made up of 3 amino acids, glycine, cysteine, glutamate, so it is also referred as, this glutathione is also referred as gamma glutamyl cystenyl glycine. You can see that all the 3 amino acids are coming here: glutamyl cystenyl glycine, that is glutamate, glycine and cysteine. Right? So it is also referred as gamma glutamyl cystenyl glycine. This is the other name of glutathione. Now coming to the third function of this glycine is the heme synthesis. Heme is made up of glycine along with succinyl-CoA. We'll discuss this heme in a separate video where we'll discuss the entire details of the heme. Then is the fourth is the purine synthesis. Purine synthesis. For that also we require glycine. The purines are made up of glycine. Now we'll discuss about the function of glycine. So the first function of glycine is creatinine synthesis. Creatinine synthesis. Creatine is made up of 3 amino acids, that is glycine, arginine and methionine. It's made up of 3 amino acids, glycine, arginine and methionine. So GAM, this is how you need to remember. Creatine is made up of 3 amino acids, glycine, arginine, methionine, GAM. The second is the glutathione synthesis. Glutathione also requires glycine. So glutathione is basically made up of 3 amino acids, that is glutamate, cysteine and glycine. So because it's made up of 3 amino acids, glutamate, cysteine and glycine, so sometimes this glutamate is also referred as gamma glutamyl cystenyl glycine. This is the other name of glutathione. Then the third function is heme synthesis. We will discuss the heme in a separate video of heme in detail. Heme synthesis requires glycine along with succinyl-CoA. Then the fourth function is the purine synthesis. We have the various purines we have, such as the adenine, the guanine. We will discuss that in nucleic acid. For that synthesis of the purine, we require glycine, aspartic acid, that is aspartate, and the glutamate. So that also requires glycine. Then the fifth function is to synthesize the hippuric acid. Hippuric acid synthesis. How it is synthesized is glycine unites with benzoyl-CoA and is going to make the glycine. The sixth and the last function of glycine is when you want to conjugate the bile acid with the amino acid, one of the amino acid that we use is glycine. So I can say for conjugation of bile acid. For the conjugation of bile acid, we require glycine and taurine. So these are the various functions of glycine we have. If you see all these 6, creatinine requires glycine, glutathione requires glycine, heme requires glycine, purine requires glycine, hippuric acid, as well as the conjugation of bile acid. All of them is going to require the glycine. Now we are going to see the details of them. So we will start with the first one, that is the creatinine. Start with the first function of glycine, that is the creatinine. How the creatinine synthesis occurs. So to synthesize the creatinine, what I can say is, first of all, glycine will join with the arginine, and we are going to synthesize a molecule that is with the name of guanidoacetate. This process is going to occur in the kidney. So sometimes they may ask that guanidoacetate molecule is synthesized where? It is synthesized in the kidney. Then this guanidoacetate molecule will reach all the way in the liver. Let's say this is the representation or the demarcation for liver. Now once this guanidoacetate will reach in the liver, what happens is, in the liver we will add the methyl group on the guanidoacetate. And earlier we have also discussed that whenever we want to add the methyl group, we will call SAM. SAM will come and will donate the methyl group. What is SAM? Stands for S-adenosyl methionine. that is going to come and going to add the methyl group. Once we add the methyl group, it will convert into a molecule that is with the name of creatine. Not creatinine, creatine. Creatine. Then on the creatine, we will transfer the phosphate group. Whenever we transfer the phosphate group from the ATP, we will take the phosphate group and will transfer to the creatine. The name of the enzyme is creatine kinase. Creatine kinase. And the creatine will convert into creatine phosphate. Creatine phosphate. And this creatine phosphate spontaneously, means without any enzyme, spontaneously will convert into creatinine. Spontaneously will convert into creatinine. So this is how the creatinine is synthesized. This is how the creatinine is synthesized. So as I told you that we are going to require 3 amino acids to make the creatinine. One is the glycine, the arginine and the methionine. Glycine, arginine, methionine, GAM, that is required to make the creatinine. Now what is the clinical importance? See, creatinine is a component of the renal function test. If you see the reports of the renal function test, there is serum creatinine, there is serum urea levels, there are the various electrolyte levels. So what I can say is, if the creatine levels is increasing, means the renal functions is getting deranged. Because this creatine is excreted by the kidneys. If it is unable to excrete, the creatine will accumulate in the serum, in the blood, and the level will rise. And that will give us an indication that the kidneys are not working properly. So this is the first function of glycine, that is to make the creatinine. What additional question that the examiner may ask is, see, we understood that to make the creatinine, we require these 3 amino acids. In the exam, they might twist the question that to make the creatine, not the creatinine, to make the creatine, how many amino acids is going to be required? You can see the answer is going to be the same. The answer is going to be the same, that is the glycine, arginine, methionine. Whether they ask creatine or creatinine, both are going to be requiring all the 3 same amino acids. Then is the second molecule, that is the glutathione. Glutathione. Let's see how the glutathione is made. We are discussing the function of glycine. The first we have discussed the details of the creatinine. Now we are discussing the details of glutathione. Glutathione is made up of, as we have written that glutathione is made up of 3 amino acids, that is glutamate, cysteine and glycine. These are the 3 amino acids which are there. Now we know that cysteine is a sulfur containing amino acid. When it comes to glutathione, there are actually 2 molecules of cysteine amino acid we have. So when 2 molecules of cysteine unites via the disulfide bond, we have discussed in the other video that where we have discussed about the classification of amino acid, we have discussed about the derived amino acid. I told you that cysteine contains sulfur. You take 2 cysteine molecules, cysteine, the 2 sulfur are there. Then the bond that is going to be there is the disulfide bond. So if the cysteine 2 molecules unite, they are going to make a molecule that is with the name of cysteine. Cysteine is going to be there. Cysteine is basically having the disulfide bond. There will be 2 sulfur groups are going to be there. Along with that, now we have the glutamate and the glycine. So I can say that it is basically a tripeptide. What is glutathione is basically, it is a tripeptide, means it is made up of 3 amino acids. It is a tripeptide made up of 3 amino acids, tripeptide. Till this point we understood everything. Now if we see the forms of glutathione, what are the various forms in which the glutathione is found? See, I can write the glutathione in this way. Now everyone just pay attention and try to understand this. See, I can represent like this. Glutamate I can represent like G. Cysteine is basically having the disulfide bond, so I can represent like S, that is the disulfide bond. And the glycine I can represent via G. So this is how we can represent the glutathione, GSSG. This is one of the way. There is one more way by which we can represent glutamate G. Now this time disulfide bond I represent via SH, and the glycine is G again. So this is how we can represent. Either we will represent by GSSG or we will represent by GSHSHG. Now, what is the difference? See, I told you in other videos also that whenever you add hydrogen to any molecule, that is called as the reduction process. If you add hydrogen, that is reduction. So, because we have added the hydrogen here in the second one, so this is called as the reduced glutathione, and the first one is obviously is called as the oxidized glutathione, because we have removed the hydrogen, that is oxidized. So if you add hydrogen, that is reduced. If you remove is the oxidized form. So we understood that there are two forms of glutathione we have. One is the reduced one and another one is the oxidized one. Till now, the glutathione we understood is having reduced or the oxidized form. It can be found in any of the way. But what we have not understood till this point is what is the use of this glutathione? Why we are discussing this glutathione at all? What is the need of glutathione in our body, right? So let's understand the function of glutathione. Let's understand there are various roles of glutathione we have in our body. So let's understand the first one. I will try to give you a diagrammatic representation so that you will have a better understanding for that. For example, let's say there is a bacteria or any foreign particle for that matter comes to our body. What the body will try to do is the body will try to kill this foreign particle, and one of the way by which the body can kill this foreign particle is by generating the free radical. Let's say this is the hydrogen peroxide free radical. The superscript, the dot at the end of the superscript is the free radical. So this is how we represent the free radical. This is the hydrogen peroxide free radical. What it will do is it will rupture the membrane of the bacteria and it will kill the bacteria or virus, whatever it is. It will kill it. At the same time, there is one vulnerable cell which is there in the circulation, and that is the RBC. This free radical can also attack the RBC because that is also present in the circulation. So this free radical will also come towards the RBC and it may rupture the RBC. So we have a protective mechanism to prevent this free radical injury to the RBC. The protective mechanism is we have a sheath on the RBC, and this sheath is made up of GSH-SHG. Means what? The reduced glutathione. So we have a protective sheath on the RBC that is in the form of reduced glutathione. Now how it is going to protect the RBC? See, reduced glutathione is having excess amount of hydrogen. So these hydrogens can be donated, and these hydrogens will react with the free radical and will convert the free radical into water. Water is not harmful. Now once this reaction is completed, what you will appreciate that now the RBC will look like this. The RBC is completely healthy and fine. But the glutathione, earlier it was in the reduced state GSH-SHG, now it will be GSSG because the hydrogens are donated. So it is the oxidized glutathione. So in order to prevent the further free radical attack, what we need to do is we need to convert this oxidized into reduced one. To convert this oxidized into reduced one, what we need to add is we need to add 2 molecules of hydrogen. And whenever we add hydrogen, that is a reduction reaction. So the name of enzyme is going to be the glutathione reductase. The name of the enzyme is going to be glutathione reductase because you are going to do the reduction reaction. If the green color arrow is the reduction reaction, means you are adding the hydrogen, is reduction reaction. If the green color arrow is the reduction reaction, the opposite, I am highlighting with the yellow color, the opposite is going to be the oxidation reaction. If the green color is the reduction reaction, the opposite is going to be the oxidation reaction. So the name of the enzyme that is going to be there when the oxidation reaction occurs, the name of the enzyme is glutathione peroxidase. The name of the enzyme is glutathione peroxidase. And this glutathione peroxidase requires selenium for the functioning. We require selenium as a cofactor for the functioning of glutathione peroxidase. So to summarize, what I can say is whenever we have free radical generation in our body. These free radical may injure the RBC. To prevent the attack of RBC, to prevent the attack of the free radical to the RBC, we have a protective sheath, and that is called as the reduced glutathione. That will donate the hydrogen, that will neutralize the free radical injury, but we need to prevent the future attack also. So that we need to replenish the oxidized glutathione back into the reduced glutathione. Right. So this entire mechanism that I have explained to you, this is called as the free radical scavenging. This is called as free radical scavenging. So this is one of the function of the glutathione that we have understood. This is one of the function that is preventing the free radical injury. This is called as the free radical scavenging. Let's see the next one. The second one is, we are discussing the function of glutathione. The second function. If we talk about the RBC, in the RBC, we have the hemoglobin. Hemoglobin contains iron. The normal iron status that is there, it is in the +2 form. +2 means ferrous. So the iron which we have in the RBC, it is in the +2 state. This is the normal form. Now, sometime it may convert into its oxidized state, that is Fe+3. That is the oxidation reaction. Sometime it may happen. And that is not normal. This ferric state is not normal. This ferric state, if it occurs and it remains for a longer duration, this will lead to cyanosis. And this is called as methemoglobinemia. This is called as methemoglobinemia. So what we need to do is, this methemoglobinemia is harmful. It will lead to chocolate cyanosis. So what we need to do is, we need to convert this oxidized or the ferric state of the iron back into the ferrous state. So that is the normal one. If I am saying that the ferrous to ferric is the oxidation reaction, Fe+2 to +3 is oxidation. The opposite is going to be the reduction reaction. So I can say this will be the reduction reaction. Reduction means you need to add the hydrogen. From where we will get the hydrogen? There is one molecule which have the excess amount of hydrogen, and that is the reduced glutathione. Reduced glutathione. So reduced glutathione will come in action and will give the hydrogen and will convert the ferric into ferrous back. Right. So this is the second function of glutathione, we can say. So let's write down: to keep the iron in ferrous state, that is Fe+2. To keep the iron in the ferrous state, that is Fe+2. Or in other words, I can say, to convert methemoglobin to hemoglobin. It will convert the methemoglobin back to the hemoglobin. Methemoglobin to hemoglobin. Or to keep the iron in the Fe+2 state. Right. So this is the second function of the glutathione. Let's see the third and the other functions. Glutathione is also required for the transport of amino acid in kidneys. See, when you transport the amino acid in kidneys, it occurs mainly by one cycle, that is called as the Meester's cycle. Meester's cycle, and that Meester's cycle requires the glutathione. So transport of amino acid is going to occur in the kidneys via the Meester's cycle, and that is going to require the glutathione. Apart from that, we can add drug metabolism, that also requires glutathione. So these are the few functions of glutathione: the free radical scavenging, then to keep the iron in the Fe+2 state, then this is the Meester's cycles, and lastly is the drug metabolism. These are the function of the glutathione. So if I just scroll up the notes and we see that what we were discussing, that there are various functions of glycine we have, right? The function of glycine that we have is the creatinine, we have discussed in detail. Then we have discussed about glutathione and their roles of the glutathione also, right? Now, the remaining ones, the heme we will discuss in the chapter heme, purine we will discuss in the nucleic acid, the bile acid we will discuss in the chapter fatty acid, right? Hippuric acid is not important, we will not discuss anywhere. So the remaining ones we will discuss in the relevant part in the relevant videos, right? So here we have discussed about the creatinine and the glutathione. Thank you.